Hi, and welcome back to the Science of Adventure. This is part 2 of a 2-video series, so if you haven't watched video 1, you may want to do that before you go on. So far, we've seen a gene get copied into RNA. Well, great, what does that do? Well, that is a copy of our recipe to make a protein, a string of amino acids, and the second process is called translation. This thing to my right is the genetic code of life, the holy grail of genetics. It took dozens of brilliant scientists in the 1960s years to figure out what all those A's, C's, G's, and U's meant. This table shows us how the genetic code is translated into protein. Every 3 letters of messenger RNA, what we now refer to as a codon, specifies one amino acid in a protein chain. Take these 3 letters, for example. That's the codon CCA. 3 bases on messenger RNA. And what they code for is the amino acid proline to be put in a protein chain. And all of these abbreviations are for amino acids. But what's really amazing about this genetic code is that it is universal. It's like a language that every living thing on the planet speaks. The codon ACG codes for threonine in bacteria, in giraffes, in tomato plants, in everything. And what that tells us is that the genetic code is probably very ancient, may have evolved in one of the first living things on planet Earth. But it's also what allows us to do gene transplants. That is, we can take DNA from one organism, transplant it into another organism, and that gene will work because all living things express genes the same way. In one of the very first experiments using gene transplantation, a firefly gene was transplanted into a tobacco plant, and the tobacco plant expressed those genes, translated them, and made a glowing protein. Before we can see translation, though, we have to look at another type of RNA called transfer RNA, or tRNA for short. I also like to think of it as taxi RNA because tRNA's job is to carry amino acids up here and bring them to the ribosome, very much like a taxi driver drives passengers around and drops them off. Also key to the function of our tRNA is this 3-letter sequence down here called the anticodon. That anticodon, as we will see, is complementary to each codon on the messenger RNA. And in the next few shots, our tRNAs are going to look like, well, little bent green sticks of dynamite. So let's begin translation. All right, first, half of the ribosome, the small half, kind of looks like a little hamburger bun, will bind to the messenger RNA at the 5' end. Again, that 5' cap on our messenger RNA may be crucial in helping orient the ribosome to that end. A tRNA will bind to the messenger RNA, and the 3 letters on that messenger RNA that start are called the start codon. The start codon is always the same 3-letter sequence, AUG. And notice up here that the anticodon UAC is complementary to that in RNA. The amino acid that's carried by that first tRNA is methionine. Then a GTP molecule will give up its energy, turning into GDP, and then the large half or large subunit of the ribosome will attach. So now the work of translation can really get going. We're going to follow it step by step. Now, in this picture, we do see that we're sort of partway through the process already. We've already formed a little bit of an amino acid chain, but let's go through all the steps so you can see how this works. First, this tRNA here is coming into the ribosome and bringing with it one amino acid. Its anticodon, which is right here, must match the codon on the messenger RNA in that same space. If it does, the tRNA will slide in there and sit right next to the tRNA that's holding our amino acid chain. Some energy is needed to make this process happen, and that will come from either ATP or GTP. But what that energy does is it helps to break the bond between the tRNA here and that amino acid chain, and then make a new bond between these 2 adjacent amino acids. So what that looks like then is this. We've basically added one amino acid to the polypeptide chain. Now, energy, again, from either ATP or GTP, will help to slide the messenger RNA through the ribosome. It's kind of like feeding a dollar bill into a vending machine. So everything will sort of shift to the left here. The tRNA that was in that spot that now doesn't have anything attached to it will exit, and the tRNA that was in this spot on the right will now move into the middle. And notice it's now holding our amino acid chain. This now opens things up for another tRNA to come in and dump its amino acid to that chain. So we build the polypeptide one amino acid at a time. So that cycle repeats, elongating the polypeptide chain, and the messenger RNA continually shifts to the left until a sequence near the 3' end comes into the ribosome. That sequence is called a stop codon. And what binds to the stop codon is not a tRNA, but rather a release factor, a molecule that looks like a tRNA. But the release factor begins the process of termination. First, the tRNA that was holding our amino acid chain will detach from the ribosome and also release the polypeptide chain. Then the release factor will trigger the two parts of the ribosome, the messenger RNA and the release factor itself, to all detach, and translation is done. So we did it. We formed a polypeptide. But if you look at most proteins, they have a far more complex three-dimensional shape than what we see in your typical diagram of a polypeptide. So in order to become functional, many polypeptides have to go through a shape-changing process we simply call folding. So that order of amino acids that we just created is called the primary structure of a protein, and it's very important. If even just one of these amino acids were out of place, the protein may not work. And if we zoom out from that amino acid chain, what we see is something like this. The polypeptide chain begins to fold on itself, but as it folds, it takes on these unique shapes. This is called the pleated sheet, and this is called an alpha helix. And you may notice that what's holding those shapes together are these hydrogen bonds, these weak attractions between hydrogen and neighboring oxygen atoms. We call this level the secondary structure of a protein. If we zoom out a little further, we see what we call the tertiary structure, or third level of folding in a protein. Now, this is similar to this little cord I have here, where I've wrapped the cord up around itself, and then I've put a twist tie on there in order to hold those folds together. So at the tertiary level, we see things like sulfide bridges and ionic bonds and even still hydrogen bonds, helping to hold kind of those coils and folds together in our protein chain. And some of these bonds are incredibly strong. The protein that makes up spider silk is so strong that researchers have suggested that a single pencil width strand could stop a Boeing 747. The result of all this folding is that each protein looks different. The unique shape we see in these three proteins is partly encoded for by the DNA that made the amino acid sequence, but it's also the way that they folded. This protein in the middle is one of my favorites. Collagen is a protein that holds down our skin to deeper tissues, and it's three of the same polypeptide braided together kind of like hair. When I was a kid, I went to a circus and I saw a guy in the circus who could stretch his skin out really far. Now, I didn't realize at the time that he had a genetic disorder that prevented linking between the collagen fibers. And I thought that if I stretched my neck out, I could actually be the same as this guy. And while I didn't get the same flexibility he has, I did get pretty far. And last, we come back to diversity. Take a look at these two DNA sequences. Can you see the differences? Let me help you. There are three. Now, these differences may not have any effect, but they may result in different amino acid orders or different primary structures, which would cause the proteins made by these two sequences to have different shapes. And so what we could be looking at here are two alleles or two different versions of the same gene. So how does the gene D4DR create an adventurous personality? D4DR codes for a protein on the surface of our brain cells called a receptor. Think of a receptor as like a baseball glove that catches baseballs. In this case, the ball is dopamine. And when the receptor catches dopamine, it activates an internal pathway that eventually leads to our sensation of pleasure or reward. Now, people with the long version or dominant allele for the D4DR gene make a defective receptor. So imagine if we took our baseball glove and cut it. When you throw a ball at it, the ball just, well, falls right out. Individuals with that dominant allele for the gene have a defective receptor that doesn't catch dopamine. The shape isn't right. And so many times dopamine comes in here, but then just passes right on. In order for these individuals to get a normal pleasure sensation, we find that they have to secrete higher levels of dopamine. And the way they do that is by seeking out more wild, crazy, adventurous activities. So if you have a friend who is always accepting any dare you throw at them or is pushing you to do crazy things with them, like jump out of a perfectly good airplane just for the thrill of it, their adventurous personality may just be in their genes. Well, we hope you enjoyed this episode of CV Biology. Remember to click the subscribe button for more lessons.